It's a true pleasure to be here today and to present to all of you. I know it's been a while. I haven't worn shoes in a talk that I've given the last 2 years, so we'll see how I hold up here. But really, it's an honor to be here, and I look forward to visiting with all of y'all, later today. And so what I'm going to do is give a brief overview, a very high-level overview of a project that we have that was funded by FAST to develop a large animal model of Angelman syndrome. And so some of y'all may be very aware of this and the work that we've done. And I kind of wanted to give you sort of a sense of where we're at with it, and then why we did this and what we plan to use it for. And so just some housekeeping things. I am the inventor of the GTX genetics antisense oligonucleotides and have interest in genetics biotherapeutics. And then, as Allison mentioned, I'm the executive director of molecular genetics at Ultragenyx Pharmaceuticals. The work that I'll talk about today was funded entirely by FAST US and also by FAST Australia, and is part of studies that we've done at my lab at Texas A&M University. So to kind of give you a high-level overview of what we'll be going over is, I want to articulate and convey to you what the role of animal models are in research. And I think that's something that's oftentimes overlooked. We just sort of talk about these things. And since there are so many animal models that exist for Angelman syndrome, I think it's important to kind of explain why we did this and how we use them and so forth. And then I'll also give you just a kind of how we made this pig model. I think it's very exciting science and very cutting edge technology that we use to do it. And then I'll show you what we've seen to date and then what we hope to do with this animal model in the future. So why have animal models for research? They play a very important, if not essential, role in what we do. And for years, we've relied largely on mouse models. That there's certain technology that existed that allowed us to genetically engineer mice, and because of that, mice became the predominant model system in all of biomedical research. In the past couple of years, we've had this advent of this new technology. I'm sure you've heard of it, this CRISPR-Cas9 technology. And so it's given us the ability to genetically modify the genomes of really anything. And so now we can make models for just, about anything, and we can use any animal to do that. And with that said, there's a lot of logic and thought that goes into which animals you pick and why you use them and so forth. And so I'll just sort of walk you through this here. We have to the best of my knowledge, I think in the Angelman syndrome field, we have more animal models than probably any condition or genetic disorder. And that's in large part due to the foresight of FAST to fund some of these models and to be thinking about how they were going to be used in the future and so forth. So we have a mouse model of Angelman syndrome. We now have a rat model of Angelman syndrome. And as I'll talk about today, is this pig model of Angelman syndrome. And so the idea here is that you can think of animal models in two sort of large areas, or two main areas. One is that we use animal models to understand the pathophysiology of the condition. So for obvious reasons, there's certain experiments that we can only do on animals, and so we can learn a lot from those animals about the condition, about the genetics, about how to manage it, and so forth. And there's a lot of information that goes back and forth. We learn from what the patient and what to look for in the animal, and then we even learn in the animals what to look for in the patient, because we can really, do a deep dive in terms of studying them and so forth. Now, the idea behind having multiple animal models is several-fold. The simplest way to put it is that there are times whenever you want to run a study, you may want to do it very quickly, you may not have a lot of money, you may want to use a lot of animals to do it, and so we rely on the mouse model to do that. It's a great animal model. We do a lot of work, and it paved the way for everything that we do, and it's usually the place that we start. We now have, as I mentioned, the rat model that's used. There are certain things about rats that make them amenable to studies, things that you wouldn't use or things that you wouldn't be able to do in mice. And so there's some thought that goes into that and some process that goes into selecting these and so forth. Likewise, we have a large animal model. And this animal model was generated primarily as a preclinical model to test therapies. So the idea was that a lot of the initial work could be done in mice and then perhaps in rats. And then as things progress towards a patient, they could be tested in a larger animal model. And this is an important part of that. So and I'll mention here in a second why we chose the pig, but one really simple way to think about this is that if you do something, let's say, for example, in a mouse, let's say you'll hear lots of new therapies that are on the horizon, lots of different technologies and so forth, and you'll hear about how they've rescued certain behaviors in the mouse, and there's all these fancy names they give them and whatnot. But a key part of that is replicating those studies before it gets into a patient. The way it used to be is that you would get this information from a mouse, and then you would have to go into a patient. One of the things that we hope to do with this, and that we're moving forward to doing, is that you could work out studies in one species and then test it in another species. And the logic is very simple. If you can cure a mouse model of Angelman syndrome, and you can cure a rat model of Angelman syndrome, and you can cure a pig model of Angelman syndrome, well, there's a pretty decent chance that therapy is going to work in a patient. So your level of confidence, your odds of success, and so forth increase as the number of species that you add to it. Importantly, though, there's certain things that you can and can't do in these different animal models, and that's where the pig model comes in. So why a pig? There's actually a lot of reasons why we chose pigs. And it turns out that pigs have actually been used as models in biomedical research for several centuries. So this is a picture here painted in 1565 about depicting studies, anatomical studies in pigs that occurred almost 2,000 years ago. And so we've been using pigs as models for a really, really long time. And there's a reason for it, and I'll kind of highlight some of those things. Pigs in a lot of ways are like mice. They breed year-round, they have large litters, we can generate a lot of animals, but there's a few key things about them that we thought would be really important for Angelman syndrome, especially given what we wanted to do and given some of the phenotypes that you see in these kids. So it turns out that pigs are incredibly smart. In fact, that some people believe that they're as intelligent as dogs. They have very complex patterns of vocalization that can represent their emotions. That's something that rodents can't do. Pigs are very unique in two ways, and that is that when they're first born, they are substantially advanced relative to other animals. In fact, pigs will open their eyes and start walking within minutes of being born. By comparison, mice, it usually takes about 10 days after they're born before they start opening their eyes. You can also wean pigs at any time. We can do C-sections in pigs and raise them in pathogen-free facilities. We can wean them the day that they're born. We can wean them all the way up until the time that they conventionally wean. And that's an important aspect of this because as many of you know, Angelman syndrome is a neurodevelopmental disorder. Many of you know that there are certain things that you see very early in infancy and whatnot. And there hasn't been a really good model for studying that. And as you likely know and will hear about, there's these different interventions, different drugs, different therapies that we're all developing. And we want to be able to test those in a way that is analogous to the way the patients will receive it, and even at the same time. And so having a large animal that is developed, is very mature when it's born, that we can isolate from its mother and do these studies on and administer these treatments is actually really important. And it's a practical aspect of this that you really can't do with any other animal, even non-human primates. It's very difficult to do these kind of studies. So how did we make these pigs? So you've heard a lot about this CRISPR-Cas technology. The easiest way to explain it is it's a molecular scissor. It's an enzyme that has really transformed the scientific field and all scientific fields. But the idea behind it is that you can design these scissors so that they can precisely cut spots in the genome. So So you can create a mutation, you can delete something, you can even add something back. And so what we did is we used the CRISPR-Cas9 technology to delete, or basically to cut out the UBE3A gene in a cell line, so the cells growing in a dish that had been isolated from a pig. So this gene's conserved in pigs. It's very similar to the human gene. We simply went in there with this technology and we cut it out. We created a deletion. It's not identical to the deletion that you'll see in Angelman syndrome, but it's very similar. So the gene is essentially gone. Then we did what's called somatic cell nuclear transfer, which many of y'all will likely know as cloning. And this is also something that's unique to pigs because there's only a few species that this is doable to date. I'd say it's argued it's more challenging than this part. But the idea is that and I've been spending too much time over here, I'll come over here. The idea is that we cut out, we have cells growing in a dish, we cut out the UBE3A gene, and then we can go in there, we can identify which ones we've created this deletion in. And then we can take those cells, those individual cells that are now missing the UBE3A gene, and we can take single cells, and then we take an egg from a pig, and we remove the genetic material from that egg. So we take a needle with a really fancy microscope with joysticks, and we suck out the DNA from that egg. And then we replace it with a cell that has been genetically modified. Then we shock the egg to make it think that it's been fertilized, and we stick it back into a recipient female, and then it goes to term, and we have a litter of pigs. And so these pigs right here, these 5 here, are the second litter that was born as part of this study. They are all identical clones of each other, and they are missing about 100,000 base pairs of the UBE3A gene, which is essentially all of it, on one copy, and then on the other copy, they have a single base pair mutation. And these guys right here were the founder animals for all the ones that we've generated today. So the way that we do this is very similar to like a mouse colony. The genetics is the same. The regulation of this gene is the same between humans and pigs. And so what we did is we have these founder males, and they were all males. They're all genetically identical. We crossed them to a normal or wild type female, to a sow. And we can control the inheritance pattern of the mutation or the deletion just simply based by the sex of the offspring. So these pigs here, and because they're pigs and because of the genetics, what happens is that you have a mixed colony. So you'll have some animals that don't get the mutation, and then you have some that do. Well, for example, this female right here, she inherited the deletion from her dad. So she's now a carrier female. And she's normal, she's unaffected because the regulation of this gene is very similar to the way it works in humans. And then what we do is then we cross this female, this carrier, to a normal male, and then she'll have one of 2 types of offspring. She'll either have pigs that have inherited this maternal UBE3 deletion, so this would be, we say, our Angelman pig, or there'll be siblings that didn't inherit that mutation, and they'll be the wild type animals. And so again, this is another advantage of pigs because of the litter size, we end up with animals that some have the mutation and some don't, and they're siblings, and so we can make comparisons between them. And these, I don't know how I go back, the picture of those were the first 3 siblings that had the deletion of UBE3A. So what have we seen and what have we done? So over the past couple of years, we've generated, I think, a little over 350 pigs. Of those, we've generated 200 that is part of this study where they will inherit the deletion from their mom. About 100 of them had the deletion, and then 100 of them did not. And so we've looked at a lot of pigs over the past couple of years. And as you can imagine, and to the best of my knowledge, this is one of the first large animal, genetically engineered large animal models of a human genetic disorder. And so there's really no textbook for this, how you raise these animals and what you do with them and so forth. So we've kind of, this has been a huge learning experience for all of us. But I'll show you some of the things that we've seen and why it was important that we did this and so forth. So we find that they have altered neonatal behavior. One of the first things we noticed in these pigs is that not all of them, and these would be the pigs that have inherited deletion, not all of them, but a substantial number of them could not suckle. And so we noticed that very early on. And so we would actually intervene. And we, because they're pigs, because we can do this, we actually bottle fed these pigs. And myself and my lab, we've all have a lot of experience doing this now, where in some instances we have to intubate the pigs, other instances we bottle feed them. But it's not all of them. It's some things that you might expect to see in patients. We see that they have very severe hypotonia. So they have very poor muscle tone. In fact, I've got a graduate student who, on the day the pigs are born, can genotype or to tell with almost 100% accuracy which one has a deletion and which one doesn't, simply by holding them. So they have this very poor muscle tone that we see that the day they're born that transitions with them throughout their life. They're very uncoordinated. They can walk, they can move around, but they have the characteristic gait where they're unbalanced, they fall, you see them moving, a little bit differently. And what's interesting about that is that transitions over time. So there's even a period whenever it's not so obvious, and then it changes again when they get a little bit bigger. We see that when they walk and when they turn, they sort of pivot. They have what we refer to as a stilted gait, where their hind legs are really stiff. They walk on their toes. Their legs are kind of close together and almost in a scissoring effect, and they, I don't know if you've ever seen pigs, but they kind of buck and jump around. These guys, they can manage, but they move around very gingerly. We also see that they don't vocalize very much, and I'll get to that here in a second, and it's very striking, and I think something that's also key about this condition, but it's evident from the day that they're born. We see that they have altered brain development and growth. It's not severe, but we do see that it's different. So there these, if you may have heard already or not, there's these deficits in synaptic connections. We've seen this in mice. We also see this in the pigs. And then we also, it's something that's unique that is not commonly found in rodents, is by chance, just pure serendipity, we see that they have spontaneous seizures. And this is a phenotype that's an important one, but this is not one that is mirrored properly in the rodent models. And it turned out we had one of the first litter of pigs that we had generated, and just by chance, they'd all come down with respiratory infection at about day 5, so these 5-day-old pigs. And every single one of the pigs that had inherited the deletion from their mom had seizures. None of their wild-type siblings did. And they actually responded to antibiotic treatment and to the same medications that you use to treat patients with. And so for us, it was just something that happened, but it's very similar to what is seen in the condition. So we don't have to induce it. It's thought to come from stress, from fevers, and we see that in this particular animal model. We also see myoclonus in the animals whenever they get older. There's a whole host of symptoms and phenotypes that we see, and almost all of them mirror the symptoms that you see in patients. Now, going back to the vocalization, this is something that I wanted to highlight because I think it's very striking. It's something we didn't expect, and kind of illustrate how we hope to use this model in understanding this condition as well as developing new therapies. But as I mentioned, we learned early on that they didn't vocalize very much. And so if anybody's ever been around pigs, raise your hand if you've ever been around pigs. I'm going to put 2 hands up. Pigs are so loud, you cannot hear yourself think when you're in a room with them. I mean, it's like torture. And like I mentioned, they communicate. They'll to exhibit emotions. So if you hold them, they'll squeal. If they're scared, they'll squeal. They'll have very high frequency squeals. If you separate them from their mother when they're young, they have a very characteristic grunt that they use that's thought that they're communicating with where they're at. They're pack animals, they're used to being together, and so there's all these stereotypic sounds that they make. And we noticed that the pigs with the deletion weren't making that. In fact, we kind of stumbled onto it when we had them all in one room one day. So we had finally gotten to a point, we had 2 litters of pigs, and We put all the pigs with a deletion in one room and all of their siblings that didn't have the deletion in another room. And there was a group of us, we walked in there, and I've spent way too much time around pigs, but I noticed, I was like, there's something wrong here. And you could have heard a pin drop in that room. And I kind of looked at everybody and I said, this is just not right. This is eerie how strange this, how quiet it is in here. And we rushed out into the other room, and pigs, when they see people, they're motivated by food. They think they're going to be fed, and they all started squealing. And again, you couldn't even hear yourself think. And it was there. It was very striking, very remarkable about how different that is, and it's important because that's an important phenotype with this condition. But I'll just sort of walk you how we're using this and what we see, and sort of illustrate what it looks like from a scientific perspective. So this is work that's done by a graduate student in my lab, Luke Myers, and he's largely responsible for all the behavioral testing that's going on with these pigs, and it really it's an amazing amount of work that he's done in terms of understanding their phenotypes and so forth. What we're looking at here is what's called a spectrogram, and basically a spectrogram is basically a way to visualize sound, just that simple. And so on the y-axis here, this is the frequency in kilohertz. So he goes from 0 to 20 kilohertz. So this is how loud, the sound is, essentially. And then on the x-axis, this is time. And so this is a 15-day-old wild-type pig that's been isolated from its mother, and it's been put into an arena. It's almost about the size of this stage up here. And as part of these studies, we look to see what their activity looks like. We look at their coordination, and then we record sounds that they make while we're doing this. And so we've got a nice system where we can do all this at once. Well, as I mentioned, so when you isolate pigs, they'll make a very characteristic grunt to communicate to their mom and to the rest of the animals where they're at. And you can see this. So it's a low frequency kind of grunt. I think I'm not going to make the sound, that would be embarrassing. But this is what you're looking at here. And you can see it's very patterned over and over again. And this is this pig walking around, exploring, looking at things, and it's making this sound. This is the sibling of that pig on the same day in the same arena, and this is the sound that it makes. And so one of the things that we've noticed is that there's actually a couple things. One is that the software that we use, the analysis that we use, we can take one of these sounds and we can actually look for it. And so if we tell the software to look for this sound, it'll find it over and over and over again. If we take this sound and we look for it here, the software can't find it. The pigs are making sounds. It's very subtle, but they can't find that pattern of sound. Likewise, if we take a sound from the pig with the deletion and we look for it here, it can't find it. So the software says that it's not detecting it. And we've had to send these off to people to try to figure out how to do the analysis. Unfortunately for Luke, I think he's going to have to do it all by hand. But nevertheless, we see that the type of noise they're making, or the way they're communicating, is different. And there's much less of it. Right? And then we see that it's different in different emotional states. So when these pigs normally would squeal, like if we're restraining them, the Angelman pigs don't do that. It's like they're not expressing the right communication in the right context. So we're trying to understand this. We were very shocked by this and how striking it was, and using this to dive into sort of to understand, if we can, what's going on here. But also, you can imagine that we can use this as a biomarker to see if a particular therapy is working. Now, these studies still need to be done, and there's a lot of work to go on. But it's a very simple way to assess a phenotype, and it's one that's very striking. And this is exactly what you want when you're looking to see if a particular drug works or not. All right, so kind of moving on, and I'm notorious for running over time, but I'm almost done. Really, why we wanted to use these is to develop new therapies. I mean, this is really what this model is intended for. Things that are promising, things that that we've seen in other model systems and other labs. Our long-term goal is to have them tested in these pigs. You can think of it as like a final preclinical stage animal model for testing your most promising therapies before they go into a patient. And this is what we're hoping to do here. And so you can imagine being used for the development of new therapies, therapeutic windows, again, because we can isolate these pigs, they're large, we can administer these drugs when they're very young, we can do it with some of the same equipment that you use in patients, and the same routes of administration that you would with a patient. And then because of their size, we can collect a lot of blood from these animals, a lot of biofluids, and so we can even use them for biomarker discovery. And I'll just kind of show you an example of what we're doing in a study that and one of the reasons I was late getting here, that we just finished up. And so this here is so Allison will be impressed by my veterinary equipment that we've got. But this is the imaging facility here at Texas A&M University, and it's right next to where we house these pigs. And so this is a Siemens CT scanner, so it's an X-ray machine. And then next to it is a C-arm fluoroscopy machine. And what you can't see, very well at least, is a 15-day-old pig that's sedated on this table that has already had fluoroscopy done on it and is about to go into the CT scanner. And so I apologize, I know it's small, but what we're doing here is we've actually injected a dye into the base of the skull, and we're watching it move throughout the CSF, and watching it go into the brain and then going down the spinal cord. And so, this is very difficult to do these. I mean, it can be done in other animals, but because of the size and because of what we can do here, it is possible and feasible in these animals. This image right here is actually taken as part of a collaboration we have with Stride Bio, testing gene therapy for Angelman syndrome. And so what I want to point out here is this would be kind of the after here, but this is a spinal needle that has been inserted in the cisterna magna, the very base of the brain. And it's got a virus, and so there's been a lot of talk of this virus, but you kind of get to see what this looks like and how you use this technology in these animals. And it's difficult to see, but that needle, the end of that needle, is right next to this subarachnoid space in the cisterna magna. So this is actually where the CSF goes around and flows down the spinal cord and throughout the brain. And the idea with a lot of these drugs, not all of them, is that they're administered either at the base of the brain or intrathecally. And so we can actually visualize this because we want to make sure that you're in the right spot when you inject this. And what's happening is that drug gets pumped or administered right there into that space. And then the fluoroscopy, they're different studies, but then you can actually see it start to distribute. So this is the kind of work that we're starting to do now that we have the animals, where we've characterized them, and now we're in parallel we're starting to test these different therapies to see if we can treat some of these phenotypes. So to kind of sum all this up, we see that the Angelman syndrome pig model mirrors many of the phenotypes present in patients, and some that we haven't seen in others. And I think that speaks to sort of one of the reasons why we did it and how we can use it and so forth. I think in my opinion, it provides a very unique model system to understand the pathophysiology of Angelman syndrome. There's a lot of studies going on. This by no means replaces any other animal model. All of these animal models are meant to synergize with each other. But we can do very unique things in these animals, and because of the complexity of their brains and the complexity of their behaviors, we can use that to try to understand things better. And then we're hopeful that this pig model becomes it is an ideal preclinical model to test therapies for Angelman syndrome. And with that, it takes a village to do this. I mean, this is a lot of work. And I think everybody here that talks, I mean, there's probably a lot of people that, even more that you don't know about behind the scenes. I hope that I've gotten almost everybody here. But if I haven't, I apologize. But there's been a tremendous effort from people at Texas A&M, from FAST, from other institutions, from the people that take care of these animals, people that help us breed these animals, the people that administer the drugs, do the imaging. It really takes a lot of people to do this. And that's, there's a lot of people that are working, for you guys to do this. And they know that. And then I want to highlight a few people, Dr. Sarah Christian, Clint Taylor, Olivia Schuler, Luke Myers, Tom Jebb, Aubrey Rinderneck, Danny Ramos, and Desiree Slaughter. These are people in my lab that, they're on the ground with these animals every day. People from the facilities, Dr. Jennifer Friedley and Destiny Taylor, they play instrumental roles. And then also the imaging facility, Dr. Vita Schutter and Julie Grandin. So without these people, there's no way we'd be able to do this. And with that, I'm only 5 minutes over. I'll take any questions, but I guess you're probably going to make me wait.
We're going to call that a miracle. You can take, if there's any questions, we'll have him on the panel as well, but I think we can have time for just a couple of questions.
So back to sort of the basics of the animal model, I understand how historically you use mice kind of as a screen, right? And if it works in a mice, then you go to the more complex animals. But can you imagine a case where a therapeutic would not work in a mouse and it would be discarded, but it may have worked in a more complex animal?
That's a great question. Yes, theoretically it's possible, but I think if the studies are done correctly, it's unlikely. But genetically speaking, there's a lot of reasons why that might be the case. I mean, there's a lot of conservation across species in terms of the gene, in terms of the way it's regulated. But, you can't ignore the fact that they're mice, they're rats, and they're pigs. And there's a reason that they're different from us, right? And so, yeah, that's the tricky one. I think, if we pull this off, really the situation you're looking for is seeing it in multiple ones. And I'm not saying that it has to be done that way. I'm just, suggesting that, if you could treat a symptom or a particular phenotype and do it in 3 different models, the odds of that working it doesn't guarantee it, but it certainly increases your chances. That's a great question.
Did you try GTX 102 in the pigs?
I think I'm not allowed to answer those questions, but I'm going to defer those questions to genetics.
Thank you.
There's one back there.
I know that you have models for the deletion and mutation, correct?
Yes.
Okay. What are the chances of you having a UPD model?
So there's in pigs, we currently don't have any plans for that. There is actually a UPD mouse model. I think I saw Dr. Baudet here. It's one of the first mouse models that was used for Angelman syndrome. I've got it in my lab. It's not commonly used, but there is a mouse model, a UPD mouse model that does exist.
Hi. How far along are you in characterizing them? And, well, I think that's sort of going to the genetics 1 or 2 questions, but when do you think they will be ready?
I think, so we have a really good, we'll continue to look at their phenotypes, but I would say that we're largely done. In terms of the phenotypic characterization at different ages, we're at the end of those studies. We have a really good sense of what to expect and what we see. I mean, there's maybe more power, like with more animals for some studies, but as I mentioned, with the numbers that we've looked at, we have a really good idea of their phenotypes. Now where we're at, as I showed in the last picture, is we're ramping up the studies to look at different therapies and then assess how those phenotypes change.